have a variety of effects on metabolism, both short and long term, and here are some of the main ones. Humans evolved to use glucose, so sugar, and fats and proteins as fuel sources. So, those processes of making and breaking them, that is their metabolism, is going to be highly regulated. Humans did not evolve to break down ethanol and use it for energy. Instead, ethanol is basically, it can be produced by fermentation of rotten things and stuff like this. So those ancient ancestors in the forest might have eaten a fermented leaf and that had some ethanol, and so the body needed to break it down. And so we have ways to break down ethanol, but that's not going to be a highly regulated process like the breakdown of those other molecules. The process of breaking down the ethanol, however, is going to produce metabolites, or these sort of chemical intermediates, and products, things like NADH and ATP, that are going to be regulators of those other highly regulated pathways for breaking down things like glucose and fats and building those up. As a result, the ethanol breakdown, even though itself is not going to be regulated, it's going to produce these things that are going to mess up the regulation of the metabolism of those other compounds. And it's going to, more specifically, we'll talk about how it's going to increase the ratio of NADH to NAD+, and this is going to cause problems that can basically tell the body that, hey, there's plenty of energy here. So stop breaking things down, stop breaking down sugars and fats, and instead store energy as fats. This can lead to things like hypoglycemia, so low blood sugar, as your liver stops sending glucose to the body because, hey, it's got enough energy, right? And it's going to cause the liver to start building up fats. It can also mess with the production of neurotransmitters. So our brains use the amino acid glutamate as a signaling molecule, and glutamate, well, it relies on products of the citric acid cycle, and we'll see how that NADH ratio can actually inhibit the citric acid cycle and therefore inhibit the production of these crucial neurosignaling molecules. So we'll talk about a variety of different effects, also things like ketoacidosis that can happen, lactic acidosis, and so we'll talk about all of these. We'll also discuss the kind of main source of those acute symptoms of ethanol toxicity that are going to come from the buildup of an intermediate called acetaldehyde that's going to be toxic to the body and cause both short and long-term effects. And so let's get into the details of all of these things. The first step of metabolizing ethanol is going to be converting it to acetaldehyde using the enzyme alcohol dehydrogenase. This is mostly going to be happening in your liver using this enzyme, alcohol dehydrogenase, but there are also other pathways for breaking down ethanol that involve the enzymes catalase or CYP450, and those can happen in different cell types as well as in different compartments of cells. And so I'm not going to go into that, but I will direct you to this really great review that a lot of this information is going to be coming from, Ethanol Metabolism, the Good, the Bad, and the Ugly, by David Wilson and Franz Machinsky, which is open access, so anybody can go and read it. And I encourage you to, and I will post a link. Okay, but going back to our story. So first we have this oxidation of ethanol to acetaldehyde. So we're converting this alcohol to a aldehyde. And then we have another oxidation step where we're going to use acetaldehyde dehydrogenase to convert that acetaldehyde into acetate. So we're oxidizing it one step further, taking it from an aldehyde to a carboxylic acid. We're then going to, in all your different tissues, can take in that acetate and use acetyl-CoA synthetase 2 to convert that acetate into acetyl-CoA. We'll talk more about this in a minute, but then this acetyl-CoA can then enter the tricarboxylic acid cycle, where it can be broken down even further, oxidized further fully, and this is going to provide a bunch of NADH. The NADH from this, as well as the NADH from these steps, can then enter the electron transport chain and be used to make energy. Going back a second to some of the chemistry, Basically, when we have these oxidation steps, we call them oxidation because one of the molecules is going to be losing electrons and another is going to be gaining electrons. So basically what's happening is that the ethanol is getting oxidized as it's reducing the NAD+. So the ethanol is transferring its electrons onto the NAD+, which generates NADH, and then the NADH can later go and cash in those electrons in the electron transport chain. Those electrons get passed from one molecule to another molecule, and each of these passings is involving something that wants the electrons less to the electrons from something that wants them less to something that wants them more. This is going to be energetically favorable, and this energetic favorability is going to be used to drive the production of ATP, which can be used as this kind of like general cellular energy source. As a result of all of this, what's going to happen is that you're going to have an increase in the concentration of ATP, a corresponding decrease in the concentration of AMP, and this is going to tell your body, hey, we're in this high energy state. One of the ways that it tells the body that you're in this higher energy state is through this enzyme AMP-dependent protein kinase, AMPK. Basically, this kinase is going to be regulated by this AMP, by this adenosine monophosphate. And so it's going to be activated by it. So if you don't have a lot of AMP, you're going to have decreased activity of this kinase. And the kinase is responsible for for regulating a bunch of different processes. Everything from mitophagy, mitochondria synthesis, glycolysis, so sugar breakdown, fatty acid breakdown, protein synthesis, all of these various things. Basically, what's going to happen is that when you have this dysregulation of these AMP levels, you're going to have this dysregulation of this AMPK levels, which is then going to cause different effects in different cells and on different processes. And so that's one of the ways that your ethanol metabolism can affect things. We'll talk a lot more later in a minute about how that NADH buildup is also going to affect things, because although this NADH can go into this electron transport chain, there can actually be a bit of a holdup to get those electrons into the nucleus, into the mitochondria in some cases. And also it's just, when you're producing so much of the NADH, it's not like it's an instantaneous thing that those electrons can be passed off. And so you're going to get an increase in your NADH to NAD plus ratio, which is then going to have inhibitory effects on various parts of the breakdown of molecules and the making of other molecules. And so we'll get much more into this, into how that NADH to NAD plus ratio is going to affect things. But first, let's go back to our story of kind of like those more instantaneous effects that you might see. Not instantaneous, but the short-term kind of like alcohol toxicity, the actual toxicity part, rather than just the dysregulation of metabolic processes, is coming from the buildup of this intermediate acetaldehyde. So acetaldehyde, remember, is coming from this alcohol dehydrogenase step, and it's before the acetaldehyde dehydrogenase step. So this ADH enzyme produces acetaldehyde, and the ALDH enzyme then oxidizes that further to acetate. Acetate, acetic acid, like your vinegar and that sort of things. That's obviously not going to be this highly toxic product. We'll talk some about how it can dysregulate things and where it enters various pathways. But the product itself is going to be like safe. It's just like an intermediate that our bodies can process normally. But this acetaldehyde, this is going to be a highly reactive compound. It's going to be like chemically reactive. So it can react even in the absence of enzymatic help. And how it can react is basically it can attack a lot of different molecules, proteins, nucleic acids, etc., with the potential to cause both short and long-term effects. You might have heard of formaldehyde. Well, formaldehyde is basically like acetaldehyde, except it doesn't have a methyl group, so it has one fewer carbon. And formaldehyde, and even sometimes acetaldehyde, we can use these in the lab as kind of like tools to do cross-linking. So basically to link proteins to nucleic acids or to other proteins to see where they're hanging out and what's near what. And one of the reasons why formaldehyde is not good for you is because it can cause these crosslinks and mess up your DNA and things like this. Turns out acetaldehyde, well, it can have the same sort of reactivity because it has that same reactive aldehyde group. So it's not going to be as reactive as your formaldehyde, but it can still cause these problems. So that is the acetaldehyde. And what happens is that basically, if the ALDH cannot keep up with the ADH, you're going to get this buildup of acetaldehyde. Some people have mutations in this ALDH enzyme that make it like slower, and this is, or have other issues. And so basically then they're going to have more of this buildup of acetaldehyde, and they get drunk more quickly, have these toxic effects more quickly. Some anti-abuse treatments, including disulfiram, what you might have heard of antabuse, what they do is they work by inhibiting the ALDH. So this leads to intense symptoms when people drink. And so it's kind of so that when people are taking this and then they drink, they get this really bad buildup of acetaldehyde, so bad that it'll make them stop drinking. Okay, so that's that. And let's talk though about, we made it all the way to acetate. Now what's going to happen? So in all of our other tissues, we can take the acetate in, and now we can use that, convert it to acetyl-CoA. When we talk about CoA, basically it comes from this vitamin B5 or this pantothenate, and this is the CoA part. It's kind of like this handle that then has this reactive thioester linkage to this acetyl group. So when you see acetyl-CoA, when you see it represented something like this is what it is actually. This is the molecule that it actually is. Sometimes you might see it with S-CoA or SH. This is basically just emphasizing that you have that sulfur linkage. Okay, so that acetyl-CoA, what are we going to do with it? One of the things that we can do with acetyl-CoA is enter the tricarboxylic acid cycle, also known as the Krebs cycle or the citric acid cycle. I like to call it the TCA because, well, that's simple. And if we look at the tricarboxylic acid cycle, what it does is it takes that acetyl-CoA, it combines it with oxaloacetate to give you citrate, and then it oxidizes that citrate all the way, producing CO2. So it basically breaks it down into CO2, and in the process produces these NADH and these FADH2, as well as a GTP or ATP. Those NADH and the FADH2 can then enter the electron transport chain to be used to power the synthesis of ATP. So that is our tricarboxylic acid cycle. And I also want you to, just a sneak peek, look, and I've shown the regulation of some of these different enzymatic steps. So in red are the things that inhibit these enzymes, and in green are the things that activate them. We can see that NADH is going to inhibit pyruvate dehydrogenase, which is what connects glycolysis to the citric acid cycle. It's also going to inhibit enzymes of the citric acid cycle, more specifically our citrate synthase and our alpha ketoglutarate dehydrogenase complexes. Okay, and so that is going to then cause a problem if we're inhibiting this tricarboxylic acid pathway. But there are additional pathways that the acetyl-CoA can be used for. And so we'll talk about how it can be used instead to generate fatty acids, as well as like ketone bodies. So that fatty acid synthesis only happens in certain tissues, including the liver. And so in your liver, where you have this most of this breaking down happening, the initial breakdown, you're building up this acetate. Not all of it is going to be sent out to other tissues. Some of it is going to be converted to fatty acids, which can lead to problems like a fatty liver. So how is all of this actually coming about? The alcohol, what it's doing is it's basically hogging this NAD+, making a bunch of NADH, skewing the oxidative balance. So normally our cells keep this ratio where we have a most of our NAD+ or /NADH, basically you switch between them by transferring a hydride, so a proton with 2 electrons. We normally keep a high ratio of NAD+ to NADH. But in this case, we're increasing the ratio of NADH to NAD plus. So basically the reverse of this. So we're making more, we have more NADH than we would normally have in order to do things like catabolism. So normally catabolism, so breaking down molecules, usually involves an oxidation, in which case we're reducing something else. So we oxidize one molecule by reducing NAD plus to give us NADH. As a result, we're going to want to have that NAD plus ready, a high ratio of the NAD plus, a high amount of the NAD plus, and less of the NADH, because we want to use it in the NAD plus form. But what's going to happen in the case of our alcohol metabolism is these two steps are both going to be using this NAD plus and generating NADH, skewing that ratio. We also, because some of that NADH can then be used to make ATP in the inoxidative phosphorylation, you're going to get this basic cellular state where your body's thinking, okay, we're doing a lot of catabolism, we're breaking down a lot of molecules, we're generating a lot of energy. We can stop breaking down other things, so stop breaking down glucose and fats, stop shipping glucose to the rest of the body, and instead build fats. So basically, you're building these same metabolites that you would build if you were breaking down things like sugar. And so you're going to be getting those molecules that tell your body, hey, stop breaking down sugar, because, well, it's supposed to be like this negative feedback that you're breaking down sugar, you're producing this NADH, which can then go and inhibit the further breakdown of sugar. In this case, we're producing the NADH without the breakdown of sugar. So at the end of this pathway, we're getting this NADH, but we're not getting the pyruvate that we would get if we were breaking down sugar. So in glycolysis, we break down glucose to give us pyruvate, which is this 3-carbon molecule. And in the case of our ethanol breakdown, we're getting this acetate, which is this 2-carbon molecule that's not going to be able to, on net, synthesize more glucose because it's only 2 carbons long. And so we'll talk more in a minute about how it influences the TCA and stuff like this. But basically, we have this product that can't be used in the same ways as pyruvate could be used. One of the things that pyruvate could be used for is it could actually be used for gluconeogenesis, so for actually making sugar. So I know this is really big diagram, but basically pyruvate is here. If we take pyruvate and we take it up, we follow these blue arrows, we can produce glucose in tissues like our liver, a little bit in our kidney, but mostly in our liver, this process of gluconeogenesis. This process relies on having pyruvate, though, and so if we don't have that pyruvate, we're not going to be able to make glucose on net, and so we're going to be inhibiting this gluconeogenesis. We're going to get less glucose produced, and this can then lead to hypoglycemia or low blood sugar. We're sending these signals like we've got plenty of energy. So instead of breaking things down further, we can actually take the acetyl-CoA and we can go and we can build fatty acids from it. To make things worse or simpler or whatever, basically fatty acid synthesis and breakdown are inversely regulated. Makes sense. If you're building fatty acids, you don't want to be breaking them down at the same time. And so alcohol is going to increase the synthesis and inhibit the breakdown. And we'll talk more about the actual details of how that happens. At its heart, it starts with this kind of altered metabolic ratios. As I mentioned, NADH is going to be a sort of feedback regulator of catabolism. So catabolism produces NADH. And so NADH is then going to signal to the body, to the cells, hey, we don't need to break down more stuff. You're good to go. And so it can act as an allosteric modulator of various enzymes. And so when we talk about an allosteric modulator, it's a molecule that binds to some part of an enzyme other than the active site, so not where the substrate binds, but somewhere else. But it in fact affects the enzyme's ability to actually process the substrate. And so NADH is going to inhibit various enzymes, pyruvate kinase, pyruvate dehydrogenase, citrate synthase, and alpha-ketoglutarate dehydrogenase. And we'll look at a second about where all of those lie in the pathway. And so then this is in addition to the fact that you actually need NAD+ in order to do those different reactions, or to do various different reactions in these pathways. So we already kind of got a sneak peek about where some of these enzymes were. So within the citric acid cycle, we have this citrate synthase and this alpha-ketoglutarate dehydrogenase complex, both of which are going to be negatively affected by NADH. So we get a bunch of NADH from the buildup, from the breakdown of ethanol. It's going to inhibit those. Additionally, we have the last step of glycolysis. So this pyruvate kinase step is also going to be inhibited by NADH, as is the step that connects the glycolysis to the citric acid cycle. So this pyruvate dehydrogenase complex is also going to be inhibited by our NADH. So again, if we go back to this big busy diagram, but now let's zoom in on the part at the bottom. If we zoom in on this, what we can see is that these steps that I've highlighted are going to be inhibited by the NADH. Additionally, we can see that what's going to happen is that if we're inhibiting these various steps, well, now we can shunt the acetyl-CoA into other pathways. So we're saying, okay, we can't take it the citric acid cycle as much. So what else can we do with it? What else we can do with it is we can do lipid metabolism. We can convert it to fatty acids, and we can convert it to these ketone bodies. We'll talk more about the ketone bodies in a second, but first let's mention the fatty acid synthesis. What's going to happen is to actually make fatty acids from acetyl-CoA, you go through this 3-carbon intermediate called milanoil-CoA. And this milanoil-CoA is then going to act as a negative inhibitor of the breakdown of fatty acids. And so we'll talk more about this in a minute, but to actually break down fatty acids, you have to get them into the mitochondria. And that step of getting them into the mitochondria is going to be inhibited by this milanoil-CoA. And so if you have this fatty acids being made, now you're going to inhibit the breakdown of these fatty acids. So that's how we inhibit the breakdown of the fatty acids, and we increase the making of the fatty acids, because basically what we're doing is we're going to be not being able to take this acetyl-CoA this way. And so instead we'll have to take it these alternative routes. So let's look a little bit more closely at one of these alternative routes, which is the formation of these ketone bodies. These ketone bodies have medical significance because, well, it can cause ketoacidosis. So if we look at the structure of these ketone bodies, so we're talking about our acetoacetate, our beta-hydroxybutirates. These are carboxylic acids, so they can acidify the blood, cause ketoacidosis, which can cause problems because, well, all the molecules in your body are going to be really reliant and specialized to work at a very specific pH, and so you don't want to mess that up. How ketone bodies form is by reversing the last step of fatty acid breakdown. So I'm not going to go into the details, but fatty acid breakdown happens in this process called beta oxidation, which is basically going to be cutting off a CoA at a time, cutting off an acetyl-CoA at a time, producing NADH and FADH2. So that's where you get the energy from, as well as that acetyl-CoA that can enter the pathway. And so this breakdown is going to happen these 2 carbons at a time. And the last step of this breakdown, you're going to be left with 2 acetyl-CoAs. This last step is going to be catalyzed by this enzyme beta-ketoacyl-CoA thiolase, or more commonly just called thiolase. And what's going to happen is if you have a lot of acetyl-CoA buildup, this last step can actually go the other direction, combine those 2 acetyl-CoAs to give you acetoacetyl-CoA, which can then be processed to make acetoacetate and beta-hydroxybutirates. Now, acetoacetate isn't very stable, and it's prone to non-enzymatic decarboxylation. So basically, it can just spontaneously lose the CO2 to give you acetone. And this acetone, it can be detected in the breath of people that have ketosis going on. So they have a lot of acetone. It can have this like fruity or nail polish remover type scent, which can kind of be used to detect that someone has ketosis. And often this is because of reasons like diabetes, where their bodies can't get enough of, their cells aren't taking in this glucose, so they can't make, they don't have enough of this products of the citric acid cycle, especially this oxaloacetate, which is needed in order for the acetyl-CoA to enter the pathway. So if we go back to our tricarboxylic acid pathway, we can see that for that acetyl-CoA to enter the pathway, it has to join with oxaloacetate. Oxaloacetate can be made from pyruvate, but it can't be made from acetyl-CoA. Because it can't be made from acetyl-CoA, for our acetyl-CoA to enter this cycle, we already need to have another source of fuel that can provide this oxaloacetate. Remember, if we did glycolysis, we'd get this pyruvate. Pyruvate could be used to make acetyl-CoA, but it could also be used to make oxaloacetate. That's an example of what we call an anaplerotic reaction, where we're basically doing some sort of reaction that's going to provide pathway intermediates to allow the cycle to keep going if we were to take things out of the cycle. And so we need basically something to provide the oxaloacetate for this process to happen. If you can't take glucose in, well, now you're not going to be able to do this process. You're not going to have enough of the oxaloacetate, and you're not going to be able to, therefore, have that acetyl-CoA into the pathway. And so this is why people with diabetes who their insulin isn't being produced, or it's not being kind of like recognized or working properly. What's going to happen then is they can't make enough of that oxaloacetate, and so they have the acetyl-CoA built up, and then it's going to cause this ketone body formation. This can also happen, however, if we don't have oxaloacetate for other reasons, if we don't have that pyruvate for other reasons, such as if we're basically just using this ethanol for the fuel source, we're not producing that pyruvate, we're only producing that 2-carbon product. We can't do any anaplerotic reactions from the breakdown of ethanol in order to keep the pathway going. And so instead, we'll have to use other fuel sources in addition to the ethanol in order if we want to get the ethanol to go through this pathway. And we're going to get this acetyl-CoA building up, not only because we have less of the oxaloacetate, but also because that NADH is going to be inhibiting these enzymes of the TCA. A quick side note that this carnitine acetyltransferase, this is the step that I said was going to be inhibited by the milanoil-CoA, which we're making, building up if we're building fatty acids. So we're inhibiting the breakdown of these fatty acids, but we're not inhibiting the formation of ketone bodies. So ketone bodies can be formed from the breakdown of fatty acids, but in this case, they're being formed directly from that acetyl-CoA. Okay, so we have this NADH, this high ratio of NADH. This is going to cause there to be more of this acetoacetate be converted to this beta-hydroxybutyrate, which is a more stable form of acetoacetate. So acetoacetate, I got it off on the sidetrack, sorry, but that led to the acetone because this acetoacetate was unstable. Beta-hydroxybutyrate is the reduced form of acetoacetate. So we take in this ketone and converted it to an alcohol. This is going to be more stable. And this is also going to be more prominent in the cases where we have this high NADH ratio. And so in patients with alcohol toxicity, say, driving ketosis, they would have a higher ratio of beta-hydroxybutyrate to acetoacetate than you might see with ketosis caused by like diabetes, where you don't have the same NADH buildup ratio. NADH can also build up for other reasons, including if you have a lack of oxygen, so anaerobic conditions. Under these conditions, you still might want to break down sugar, so you still want to do glycolysis, but you're going to be building up the NADH, and we talked about how the NADH is then going to inhibit various things, as well as you're going to need more NAD plus in order to keep the cycle going. Fermentation is going to provide ways to regenerate the NAD plus without oxygen, so glycolysis can continue anaerobically. So this is how ethanol is actually made by some yeasts and plants and microbes. So you get that alcohol made that you can then drink. But in our bodies, we do lactate fermentation, where we can convert pyruvate to lactate. That lactate can then be transported from the various tissues that are producing it to the liver, which can then convert it back into a useful form, convert it to pyruvate, use it to make glucose that it can then ship back to the muscles and the other cells. But in the case of our ethanol metabolism, so you might see some like lactic acidosis because you have this high NADH ratio that is then going to be converting the pyruvate that's present to lactate. But you're not going to see like a huge buildup of things as much as you would see with under conditions of no oxygen because, well, you don't have that much pyruvate to begin with. So you're going to get this altered ratio of lactate to pyruvate because you're going to have more of this NADH driving the conversion of pyruvate to lactate, but you don't have that much pyruvate to begin with. So you don't have that much pyruvate to begin with. Now you have even less of it. And so what's going to happen is that's going to make it harder for you to do things like gluconeogenesis to make glucose from that pyruvate, or to have enough pyruvate to make oxaloacetate to keep that pathway going, to keep the tricarboxylic acid pathway going, various effects like this. Increased acetyl-CoA can also alter epigenetic regulation. So when we talk about regulating gene expression, like when different genes are used to make their proteins. So when do we make proteins from this gene versus proteins from this gene? There are various levels which we can regulate things at. And one of them is the level of transcription. So actually making a messenger RNA copy of that DNA gene that can then be used by the ribosomes in the process of translation to make a protein. In the process of transcription, so we're making this mRNA copy, it's going to depend on things like how accessible the regions of the DNA are to the RNA copier, so this RNA polymerase. They can be inaccessible because they're wound up around proteins called histones. And you can then, however, this keeps things nice and tight and tightly regulated. So it allows all the DNA to fit snugly inside of your nuclei, and it also makes it so that your cells can regulate when various regions are accessible. One of the ways that they regulate this is by regulating the proteins, those histone proteins that the DNA is wound around. These histone proteins have a bunch of these lysines, and these lysines can get modified in ways including methylation and acetylation. And it's this acetylation in particular that I want to talk about here, because this is actually going to involve attaching an acetyl group from acetyl-CoA. If we have a lot of acetyl-CoA building up, such as we would have with this ethanol metabolism, well, now you're going to potentially have increased acetylation, which can then cause effects on gene expression, because it's allowing transcription factors to basically access different regions of genes. And so this can then have various both short and long-term effects on gene expression. Your body can also have various effects on gene expression in order to compensate for the changes in metabolism that are occurring. So if you're drinking a lot of alcohol, most of your energy source can actually be coming from that ethanol as opposed to other energy forms. And so your bodies could then regulate, like downregulate the enzymes in various molecules necessary to break down things like glucose and proteins and lipids, and instead focus on making the enzymes that are needed to deal with the ethanol. And so then if you take away the ethanol, you can get withdrawal symptoms, both short and longer term. And so basically your body is then getting used to using the ethanol. So like, okay, this is the way we're going to do things. Okay, I'll adapt. And then if you take away the ethanol abruptly, well, now your body's like, whoa, now what do? I wasn't expecting that. And now I need these other fuel sources. And so that's why you can kind of get, you might have heard of like hair of the dog and things like this, where people say like, They drink a little bit of alcohol if they're hungover. Basically, it's kind of easing your body back into using other fuel sources. And I am not advocating any sort of alcohol drinking, hangover reduction, any sort of thing like that. I'm just talking about the biochemistry that's going on. And you can all drink responsibly, hopefully, because alcohol can have a lot of different effects, which can be very bad. The effects can be different on different tissues. So we talked a lot about what's going to happen in, say, your liver and your muscle, but what about your brain? Your brain, it relies on glutamate synthesis from the products of the citric acid cycle in order to produce these, to use glutamate as a neurotransmitter. So glutamate acts as kind of like an inhibitory neurotransmitter. So it's going to kind of like calm down various processes and cell types and signaling. So what can happen is that you can, with the ethanol, what it can do is it can actually decrease this glutamate production, decreasing the inhibitory signaling. It can also increase dopaminergic signaling. Kind of like how we talked about before, how your body can adapt to use various metabolic fuel sources more than others. Well, they can also adapt to say, okay, well, we know that there's not going to be as much glutamate, so let's get more sensitive to the glutamate that there is. Let's put a bunch of receptors and that sort of thing. Let's remove some of those receptors for those other molecules. And so you can therefore get these changes in the various receptors in the brain and how sensitive they are and various things like this. I'm not a neuroscientist. I'm not going to get into it, but you can go check out this article if you want more. What I will talk about briefly is how it's actually causing these effects. Well, if we look at where we get glutamate from, so glutamate's this amino acid, and so we can use it to build proteins, and we can get it from breaking down proteins. But in our brain, it's actually relies on making it from alpha-ketoglutarate. So in our brains, you make it from this alpha-ketoglutarate, which is one of these intermediates of the citric acid cycle. And we talked about how the citric acid cycle is going to be inhibited. So we're going to be inhibiting the making of this glutamate. And so glutamate can be made just by basically taking the amino group off of, adding the amino group from various amino acids onto alpha-ketoglutarate to give us glutamate. Glutamate is a lot of different uses. It's actually one of the ways that we get rid of free amino groups that we don't want and safely. And it's also going to be used in the brain as this neurotransmitter. And so now the ethanol metabolism can be messing with that. So to review, some of the effects of alcohol metabolism are it produces this buildup of acetaldehyde, which is toxic. Remember, think of your formaldehyde. You don't want formaldehyde in your body. You don't want acetaldehyde in your body. It's also going to increase the NADH to NAD+ ratio, which is going to inhibit pyruvate dehydrogenase in the citric acid cycle. But it's going to produce acetyl-CoA, and so acetyl-CoA is going to build up. This acetyl-CoA can get converted to ketone bodies, which can cause ketosis, and it can affect epigenetic acetylation, which can alter gene regulation. It can also get converted to fatty acids and cause like fatty liver. It can get triglyceride formation and things. Additionally, that high NADH ratio is going to cause the conversion of pyruvate to lactate, which can lead to lactic acidosis and further decrease the amounts of pyruvate that you have. All of that can decrease the production of glucose, so your gluconeogenesis. So your liver is not going to be providing glucose as much to the rest of the body. So you might get hypoglycemia or low blood sugar. Alcohol metabolism can also decrease the amount of AMP, which downregulates the AMP kinase, the AMP-dependent kinase, AMPK, which has a lot of different effects. It can also alter glutamate production, dysregulating neurosignaling. So I hope that helps you understand basically how ethanol is broken down in our bodies, some of the problems that can happen with heavy consumption of alcohol, both short and long term. If you want to know more, I highly recommend this review article, and please drink responsibly if you decide to drink.